Hey everyone, and welcome to another deep dive. You ready to unravel the mysteries of brain wiring?
Absolutely, I am.
Awesome. So we're driving in some fascinating excerpts from a book chapter on developmental neurobiology.
Okay.
Really trying to understand how all those billions of neurons in our heads manage to connect in such a precise way.
It's mind-boggling, isn't it?
It really is. And, this is about way more than just geeking out over science.
Oh, yeah.
It gets to the core of what makes us, us.
Right, like all our thoughts, our emotions, even our memories, they all rely on how our brains are wired.
Exactly. So to kick things off, let's go back to the mid-20th century with Roger Sperry, this pioneer in neuroscience.
Okay.
He came up with this revolutionary idea called the chemoaffinity theory.
Right.
Basically, it says that neurons have these unique chemical identification tags, almost like little addresses, guiding them to the right spots in the brain during development.
Like a microscopic postal service.
Yeah.
Sperry's theory was a total game changer.
Yeah, before that, people thought brain wiring was more random. But Sperry's theory suggested this crazy level of precision.
Totally. It sparked a whole new wave of research.
And of course, we can't just take Sperry's word for it, right?
Yeah.
We need the evidence.
Of course.
And that's where Friedrich Bonhoeffer comes in.
Okay.
Now, this guy was originally a physicist.
Wow.
Can you imagine? Switching from physics to biology. Talk about a career change.
That's quite the leap.
He designed these really elegant experiments to actually try and find these guidance molecules Sperry was talking about.
Interesting.
He created these in vitro assays, basically giving neurons a choice.
A choice.
Yeah. Like, do you want to grow towards this tissue sample or that one?
Clever.
What he found was amazing. Neurons from different parts of the retina actually showed preferences.
Preferences?
Like they were choosing their paths based on some invisible signal.
So they weren't just growing randomly.
Nope. It was like he built a tiny neural GPS in a petri dish.
That's incredible.
But here's the twist. The discovery of ephrins and their receptors completely shook things up. These were some of the first guidance molecules ever discovered.
Okay.
And they were found in the visual system, Sperry's main area of expertise. But here's the thing. They weren't attractive cues at all. They were repulsive.
Repulsive? What do you mean?
Instead of guiding neurons directly to their targets, they were actually pushing them away from certain areas.
So instead of finding the right address, it's more about avoiding the wrong ones.
Exactly. It added a whole new layer of complexity.
That's fascinating.
Bro.
So how does this fit with Sperry's original idea then? It seems like ephrins throw a bit of a wrench in the works for chemoaffinity.
Right. That's where Michael Gaze comes in.
Okay.
He proposed this idea that maybe neurons use relative positioning and competition to make those precise connections.
Relative positioning.
Yeah. Instead of having a specific street address for each house, it's more like the third house on the left kind of thing.
Interesting. Especially in crowded areas like the cortex, that would make a lot of sense.
Exactly. It allows for flexibility during development.
So does all this mean we just toss out chemoaffinity completely?
Well, not quite. It seems like there's still some truth to it, even with these repulsive cues and relative positioning in the mix.
Okay, so how do we reconcile these different ideas then?
It's about refining the concept of chemoaffinity.
Refining.
Yeah. We could differentiate between Sperry's strict chemoaffinity, where every single neuron has its own unique molecular ID,
Okay.
and a broader concept, general chemoaffinity.
General chemoaffinity.
In this view, molecules play multiple roles during development.
Multiple roles, like what?
It could be attraction, repulsion, even just creating a supportive environment for growth.
So it's not just about guiding them to the right spot. It's about making sure they can actually grow and thrive.
Exactly. Think of it like this.
Okay.
You need street signs to navigate, race. But you also need oxygen to breathe.
Right.
One guides, the other enables.
Okay, I'm starting to see the difference.
So even if ephrins aren't those super specific address tags, Sperry imagined they're still shaping the brain's landscape in a big way.
They're playing a crucial role, even if it's not exactly what we initially thought.
But this brings up a really important question.
What's that?
How can genes possibly encode the sheer complexity of brain wiring?
It's true. Billions of neurons, trillions of connections. How do you even begin to explain that with DNA?
It's like trying to fit the entire Library of Congress onto a single flash drive.
Way too much information. So how do we even approach this problem?
That's where the idea of algorithmic growth comes into play.
Algorithmic growth.
Instead of trying to hard code every single connection, maybe genes provide a set of rules and instructions, allowing the brain to wire itself up through these dynamic interactions.
So it's more like a flexible blueprint that can adapt as the brain develops.
Exactly. And to really dive into this idea, we need to shift gears and look at a simpler system.
Simpler.
Yeah. That brings us to Sidney Brenner and his groundbreaking work with this tiny nematode called C. elegans.
C. elegans? What's that?
Well, it's a worm, but don't let its simplicity fool you.
Yeah.
It has a nervous system that's taught us a ton about how our own brains work.
A worm. Okay, you've got my attention.
We'll save that fascinating story for part 2 of our deep dive.
I'm intrigued already.
Welcome back to the deep dive. Before we jumped into Sidney Brenner's world, we were talking about netrin.
Yeah.
This guidance molecule that seemed like the perfect example of Sperry's chemoaffinity theory at work.
Right, it seemed to be attracting axons to their targets just like a magnet.
But science loves to keep us guessing, doesn't it?
It does. The more we learned about netrin, the less straightforward it became.
So what happened? Did those tiny magnets stop working?
Well, imagine this. Scientists removed netrin from its supposed target area in the developing spinal cord.
So if it really was the main guiding signal, you'd expect those axons to go crazy, right?
You'd think so, wouldn't you?
Totally lost without their beacon.
But that's not what they found at all.
What? They still found their way even without netrin?
That's right. It turned out netrin's presence along the axon's growth path was far more important.
So it's not like a lighthouse beaming from a distance, more like trail of breadcrumbs.
That's a great analogy. Instead of one big aha moment, it's a series of smaller confirmations along the way.
Like, okay, I'm on the right track.
Okay.
Still good.
Exactly. It's the journey, not just the destination.
Even for neurons.
This finding really changed how we thought about netrin.
So it's less like a precise address code and more like a support system.
Yeah, more like a scaffold guiding them in the right direction.
But not necessarily micromanaging every step.
Right. And this hints at a bigger picture.
A bigger picture.
It's not just one molecule calling the shots.
Okay.
We have this whole interplay of signals happening.
Interplay of signals?
Other molecules, the physical properties of the surrounding tissue, even the timing of development. All these things matter.
So it's a team effort.
It really is. A symphony, not a solo.
Wow, when you put it that way, those billions of neurons and trillions of connections seem even more amazing.
They really are. And this brings us to the concept of composite instructions.
Composite instructions.
Instead of one molecule dictating the whole journey, we have multiple factors working together, each contributing a piece of the puzzle.
Like a recipe. You need flour, sugar, eggs, all these ingredients working together.
Perfect analogy. No single ingredient makes the cake on its own.
And this idea of composite instructions helps explain why removing one factor,
like netrin from its target,
doesn't necessarily lead to disaster.
Right. The system has backup plans.
Backup plans for the brain. That's reassuring.
It's built-in redundancy. Other cues can step in and compensate.
Okay, so the brain has a plan B. And a plan C, and probably a plan D too.
Most likely. But this makes things even more complex to study, doesn't it?
It does. How do scientists even begin to untangle this web of interactions?
It's a big challenge. We need to shift our perspective.
Shift our perspective how?
We can't just focus on individual molecules.
Okay.
We need to see how they all interact.
So it's like trying to understand a city by studying individual buildings. You need to see the traffic flow, how people move, how different neighborhoods are connected.
That's a great way to put it. And it brings up another question. What's up? Where do we draw the line between instructive and permissive roles?
That's a good one.
What seems passive in one context might be actively guiding development in another.
It's all about context.
Exactly. It's like saying oxygen is just permissive for driving.
Okay.
Sure, you need it to breathe, but it doesn't tell you where to go.
But imagine driving through the mountains where the air is thin. Suddenly, oxygen becomes a lot more instructive.
That's a perfect example, and this highlights a really crucial point.
What's that?
Information isn't just encoded in individual molecules.
Okay.
It's in the relationships between them, their spatial arrangement, their timing.
It's like a language.
Yes. The meaning comes from the order of the words, not just the letters themselves.
So we're not looking at a static blueprint, but a dynamic process.
Like a choreographed dance.
Where every step matters, but it's the timing and coordination that really create the magic.
Now remember Sidney Brenner and his quest to understand how genes build nervous systems.
Yes, the worm guy.
He knew tackling the human brain head-on would be nearly impossible.
Too many neurons, too many connections, way too complex.
So he chose a simpler model. The tiny nematode C. elegans.
Ah, yes. The worm. But why a worm?
Well, C. elegans has a nervous system with only 302 neurons.
302, that's it?
That's it. Compared to the billions in our brains, that's nothing.
Because it's like a starting point, a simpler model to help us understand the basics.
Exactly. Brenner had this audacious goal.
What was that?
He wanted to map every single neuronal connection in C. elegans.
Every single one.
To create a complete connectome.
Wow.
Yeah.
So did he actually manage to do it? Map the entire worm brain?
He did. After years of painstaking work, the first complete connectome was published in 1985.
1985. That was a while ago.
It was a monumental achievement.
Like having the wiring diagram for a simple machine.
It was a huge step forward.
So did this worm wiring diagram unlock all the secrets of brain wiring?
Well, not quite. As Brenner himself later admitted, the connectome didn't tell the whole story.
So it was a piece of the puzzle.
Yeah.
But not the whole picture.
Right. It was like having a detailed map of a city without knowing the language or the culture of its inhabitants.
You need more than just the map to understand a place.
Exactly.
Welcome back to the Deep Dive. We've been on this incredible journey exploring chemoaffinity, from Sperry's initial vision to the much more intricate picture we have today. Last time we were talking about Sidney Brenner and that amazing worm brain map he created.
Right, the complete connectome of C. elegans.
It was a landmark achievement, but like you said, it didn't immediately reveal all the secrets of brain wiring.
It gave us the structure, but not the whole story.
So how did Brenner go about trying to understand those missing pieces?
Yeah.
You know, the how of brain wiring.
Well, Brenner was a pioneer of this approach called forward genetics.
Forward genetics.
His idea was, if you want to understand how genes work, mess with them and see what happens. Mess with them. Yeah, he induced random mutations in C. elegans genes.
So basically causing little errors in their DNA.
Exactly. And then he looked for interesting changes in the worm's behavior.
So it's like randomly tweaking things and seeing what breaks.
It was a bold strategy, but it led to some really important discoveries.
What kind of discoveries?
One of the most common things he observed was uncoordinated movement.
Uncoordinated movement.
Yeah, worms that couldn't crawl in a straight line.
Interesting.
He called these UNC mutants.
UNC for uncoordinated, I guess.
Exactly. And it turned out that some of these UNC mutants had problems with their brain wiring.
So a single gene mutation could mess up their ability to move.
It highlights just how important genes are for development.
And did they figure out which gene was causing these wiring problems?
They did. And guess what it led to the discovery of?
I don't know. Tell me.
Netrin. The very molecule we were talking about earlier.
Netrin again. But we were just discussing how it's not just a simple address code.
I know, right?
Right.
It turns out netrin plays a much more complex role than we initially thought.
Okay, so spill the beans. What's the deal with netrin?
Well, yes, it can act as a long-range attractant,
Okay.
drawing axons towards a target.
Like we originally thought.
But it can also act over shorter distances. And it can even be repulsive in certain situations.
Whoa, repulsive.
That's a twist.
Plus, it interacts with a whole bunch of other molecules.
So it's like a multitasking master of disguise.
That's a great way to put it. It all depends on the context.
Context is everything.
You got it. We can't just look at a single molecule in isolation.
Okay.
We need to consider the whole network, the timing, the other players involved.
It's all about teamwork.
And that brings us back to Brenner and his worm brain map.
Right. How does netrin's complex role fit into all of this?
It shows that even in a simple nervous system like C. elegans, the wiring process is incredibly dynamic. It's not just a straightforward follow the instructions kind of thing. So we've gone from this neat and tidy address code idea to this much more intricate dance of molecules and interactions.
It's amazing, isn't it?
It really is. It's humbling to think that something as complex as our thoughts and emotions emerges from this incredibly intricate dance of molecules.
And it raises a really fascinating question.
What's that?
If brain wiring isn't solely determined by our genes,
Okay.
then what role does experience play?
Ooh, that's a good one.
How do our interactions with the world, our learning, our memories, all those things shape our neural connections?
That's a question worth pondering. That's for sure.
It reminds us that there's still so much to discover about the brain.
And that this journey of exploration is far from over.
Absolutely.
Well, thanks for joining me on this deep dive into the world of brain wiring. It's been a fascinating journey.
It really has.
And to all our listeners out there, thanks for tuning in, and